All right, hello everybody. We're continuing on with our story here. We're still trying to get our muscle fiber to contract. And so the next part of the story is, how in the world do we go from having an electrical change across the surface of the muscle fiber, that's excitation, how in the world does that lead to the muscle fiber finally contracting? And so this part of the story is called excitation contraction coupling. And again, all that really refers to is the sequence of events by which an action potential on your sarcolemma, how does that lead to the sliding that we took a look at earlier, where your actin and your myosin fibers come together and they start doing that cross bridging and they slide across each other. What's going to happen is, if you guys remember from your muscle fiber microanatomy, you've got these little tubes that are really just portions of the sarcolemma, the cell membrane of the muscle fiber, that invaginate and go down inside the muscle fiber and form a little membrane covered tube that goes down inside the cell. That's called a T-tubule. And the action potential is going to spread down those, and that is going to trigger calcium ions to come out of your SR. SR stands for sarcoplasmic reticulum. Those are membrane covered compartments where calcium ions are stored. Saw a little bit about that earlier. And here's a blow up reminding us about what's going on here. All right, so over here we've got an axon of a motor neuron, and there's our neuromuscular junction right in there. There's your ACH. Your acetylcholine came across the synaptic cleft, and that triggered an action potential to spread all the way across the sarcolemma, the membrane of our muscle fiber, and that is going to actually spread down wherever you have those T-tubules. They're only showing you part of one there. It's going to spread down those T-tubules inside the muscle fiber. And go back and review if you don't know what all these things in here are. You need to go back and review your muscle fiber microanatomy and become familiar with all of these parts we have in there. Okay, so here we're blowing up in more detail what's going on. We've got this action potential traveling along the sarcolemma. Okay, and then this is a T-tubule. So that's really just part of the cell membrane there that is proceeding down internally into the muscle fiber. All right, and as that happens, there are proteins sitting there in the membrane that respond to that change in voltage, and they cause calcium channels to open. That's what they're showing you in 4 different locations here on the diagram. So that action potential spreading down the T-tubules causes calcium channels to flip open. Those calcium channels are part of the SR, or sarcoplasmic reticulum. And your sarcoplasmic reticulum, again, stores calcium ions. So you got calcium ions stored inside that compartment. Okay, so our action potential has triggered calcium to come out of the SR. And so now it's going to, those calcium ions are going to float around all inside the intracellular fluids of our muscle fiber. And then they're going to be able to come into contact with your actins and your myosins and all those proteins that are important for muscle fiber contraction. All right, so if we continue on, now we're going to go back to the actin and the myosin filaments, our thin and our thick filaments. So if it's been a while since you saw those video lectures, you might want to go back and review all those internal parts. All right, so remember, our actins and our myosins are all bundled up in those myofibrils, and you have tons of those extending all throughout the inside of your muscle fiber, so they're all over the place in there. And if you remember from, oops, go back over here for a second. When we talked about actin and myosin earlier, your thin filaments are composed of lots of actin. Those are the little blue bead-like proteins you see here. And then you've got the tropomyosin ribbons. And then you've got these yellow proteins that are drawn in there called troponin. Tropomyosin, those ribbons, they block. What they do is when a muscle fiber is relaxed, tropomyosin blocks the active sites or the binding sites, the contact sites between your heads on myosin and the actin. We need those heads on the myosin to come into contact with the actin, those little binding sites on actin, in order for our cross-bridging to take place. All right, so what happens when calcium is around, when you have calcium levels high enough in your intracellular fluids, calcium binds to the troponin, the yellow protein that you see there, and it causes the troponin proteins to change shape, and they actually pull the tropomyosins, those ribbons, away from the binding sites on actin. Okay, so those binding sites kind of look like olive pits there. Okay, so once that happens, now the myosin heads are going to be able to bind to actin. They're going to be able to come together. They'll make a nice little fit with each other. Okay, so and you can see that's what they're showing you here in the diagram. Once tropomyosin slides away, the troponin, calcium binds troponin, causes troponin to change shape. Troponin slides the tropomyosin away from those binding sites on the actins, and now the myosin can bind the actins. You're going to be able to have that connection between the two, or that cross-bridging that takes place. So all of that, in order for any of that to happen, guess what you've got to have? The key thing is you've got to have lots of calcium ions out in your intracellular fluids. So backing up, what triggered that? What triggered that was those calcium ions had to come out of the sarcoplasmic reticulum. Well, what made that happen? Calcium channels had to open up to let them out. What made the calcium channels open up to let them out? It was that action potential that was spreading across the sarcolemma and down into the T-tubules. Well, what made that happen? Well, you had that signaling at the neuromuscular junction between the motor neuron and the muscle fiber. Where did that signal come from? Well, the motor neuron had to generate its action potential, all that earlier part of the story. So we're finally getting down to the end here. All right, and so this slide is just reviewing for you what I talked about already. If you have a low intracellular calcium concentration, so you don't have very much of it around, those tropomyosin ribbon-like proteins block the active sites on actin. Your myosin heads cannot attach to them. And in that case, your muscle fiber is relaxed. That cell is not contracting. But when you have all that calcium that comes rushing out of the sarcoplasmic reticulum, calcium binds to troponin, troponin changes shape, slides your tropomyosin away, and then that is going to start the events of the cross-bridge cycle. Now our actins and our myosins are going to be able to make that connection, and they have to detach, make another connection, detach, make another connection. That's our sliding filament part of the story, which we talked a little bit about earlier. I've got another animation link there, and again, I'm going to have all of those for you in Blackboard. So the next lecture, now we're finally, our actins and our myosins, finally they're able to get together, so we're going to review what's needed for that sliding to occur, that cross-bridge cycle to occur, because that's what's actually going to lead to the contractions that we experience in our muscles.